Hello, everyone. My name is Roman Huszár, and I am a fourth-year PhD student in the Buzsáki lab at NYU. And I'll be talking about how developmental forces shape the activity patterns in the hippocampus today. And as many of you probably know, the neurons in the hippocampus fire at discrete locations in space. These are the so-called place cells. And much research has looked at how the activity patterns of these place cells during an experience come to then be reflected in offline states during sleep. After the experience. This is the so-called replay phenomenon, and it's one that really emphasizes the plasticity and flexibility of the hippocampal system. And there's also this competing observation, which is claimed that sometimes the activity pattern during an experience can already be detected in sleep prior to the experience itself. So this is the so-called preplay idea, and it's one that really suggests that the activity patterns in the hippocampus might to some extent reflect pre-existing dynamics. And here I'm going to ask where these pre-existing dynamics may come from. And we are pursuing this hypothesis that the network dynamics are shaped by the processes that give rise to the circuits themselves during development. And so during embryonic development, the rate at which neurons are born display this bell-shaped nonlinearity. And what we'd like to do is record from neurons across this distribution and study their properties. And so to do that, we'll use this technique called in utero electroporation, in which we inject a plasmid into the ventricle of a developing embryo, and then we pass current in order to direct the DNA into the progenitors that give rise to a brain region of interest. So in our case, that's going to be the CA1 subregion of the hippocampus. And after that, once the animals develop normally and become adults, we'll implant them with silicon probes, and we'll perform optical tags in order to identify these birth-dated neurons. And we'll be electroporating channelrhodopsin and TV tomato. And so here you see an example image shows a sparse labeling in a subpopulation of neurons that we transfected at embryonic age 16 or E16. And when we record from these animals, we give 2 millisecond light pulses, and we look for reliable firing after the pulse, in order to identify neurons that had been birth dated in this fashion. And what you can see here is that as a function of different birth dates that we've done, we see that we label proportion of pyramidal neurons that reflects this nonlinear rate of neurogenesis that I showed 2 slides ago. So that's a nice sanity check that this method is working properly. And one thing that we really were curious about is if there is any effect of birth date on the correlated firing of neurons. So what we'll be looking at here is if there's any systematic difference in the pairwise correlations of neurons that were co-birth dated in this fashion. And we'll be performing this analysis inside sharp wave ripples, which is when many of the neurons are discharging spontaneously. And what we see is that for pairs of neurons with the same birth date, co-optotagged, the pairwise correlations are higher, and this effect depends on the anatomical distance of these birth dated neurons. So the ones that are nearby have the strongest pairwise correlations. And of course, we were very interested to see if this bias in correlated firing would manifest itself when the animals are navigating, when you see these place cells, so at discrete locations in space, you see that there is reliable firing trial by trial. And what we are going to do here is for neurons that were co-birth dated in this fashion, we'll look to see if the overlap, if their firing rate maps in space is higher than between neurons that were not birth dated in this fashion. And indeed, the pairwise correlations of the rate maps are marginally higher. And again, this effect is most strong between co-birth dated neurons that were nearby in anatomical space. So this kind of leads us to propose this idea that perhaps there is topography in the hippocampal representation if you condition on this variable of birth date. And so in my poster, I'll be developing this idea quite a bit further. And I'll also be talking about how the pairwise correlations at different birth dates give rise to higher order correlated states that are unique. And I'll speculate about how the differences across birth dates of these higher order dynamics might give rise to differences in single cell level properties, such as the number of place fields that a neuron might generate. So if that's something that you find interesting, I'll be happy to talk to you more about this in poster session 3. And thank you for your attention, and I'll talk to you there.